Hey y'all, listen. One of the most important distinctions between different tissues and even between different cells in the same tissue is their receptor complement. It's what receptors individual cells express that determine their fate during embryonic development. And I'm not going to lie, we are barely going to scratch the surface of cell signaling here. If you take cell biology or you've already taken cell biology, you either will or have spent a couple of weeks talking about cell signaling, and that still just scratches the surface. Way back in the Stone Ages, when I was in graduate school, I had to take an advanced molecular biology course because I was getting my degree in cell and molecular biology, and we spent nearly half the semester on signaling. So it is heckin' important. We don't have time to get into all the details of it right now, but at least let's kind of get the broad strokes of it so that when we use some of this terminology later on, you'll know what the heck I'm talking about. So there are basically three main kinds of signaling. So let's imagine this is our cell. So this box here is our cell, there's its nucleus, and the little star in here is the signaling molecule. And any molecule that will bind to a receptor, here indicated by an arrow, is referred to as a ligand. Now, you might have heard other professors say ligand. Both of those are correct pronunciations. It happens to be that the guy I learned cell biology from went to school in England where they say ligand. If you learned cell biology from somebody who learned it from somebody who's more American, then you say ligand. Both are correct pronunciations, but Dr. Edwards taught me ligand, so that's what I'm stuck saying. Although you'll still probably hear me say ligand sometimes. Anyway, I digress. So there's three different ways that one cell can signal some other cell, or even, in this case, itself. So this cell is going to release a signaling molecule, a ligand, that binds to a receptor on the surface of that same cell. We refer to that as autocrine signaling. Sometimes the cell that's secreting the signal, in this case our cell on the left here, and it's going to be sending that signal to a very nearby cell. So it's just going to zip right up here to this next door neighbor cell. We call that paracrine signaling. Now, there is a special kind of paracrine signaling that takes place only in these special junctions between nerve cells and the cell that they're communicating with, and we call that synaptic signaling. And we're going to get a little bit more into that when we get into nervous tissue, but for now, suffice it to say that whenever you're talking to somebody really close by, that's a paracrine signal, and then synaptic signalings, we're going to consider a special kind of paracrine signaling. And then finally, sometimes the cell that needs to receive the signal is some great distance away from the cell that's going to be creating the signal. An example would be, let's say that cell A on the left here is in the pituitary gland, and cell B on the right here is in the ovaries. So if this is, say, follicle stimulating hormone, which is just one example of a signaling molecule, then the pituitary gland would release that into the bloodstream, and it would travel through the bloodstream to the ovaries, where it would bind to its receptor on the surface of the cell and cause some change in the physiology of this downstream cell. When you have this kind of signaling between two very distant cells using the bloodstream as the intermediary, that's called endocrine signaling. So one cell can signal itself or another cell via autocrine signaling, paracrine signaling, or endocrine signaling. And those signals come in two main varieties. So there's two ways to signal another cell. One is using some sort of lipid. In this case, our example are steroid hormones. And lipids, because they're hydrophobic, can freely dissolve through the plasma membrane, bind to an intracellular receptor directly, and affect the physiology of that downstream cell. But hydrophilic signaling molecules, hydrophilic ligands like these protein hormones, they can't get through the plasma membrane because they're hydrophilic, and that hydrophobic barrier of the plasma membrane prevents it. Instead, they're going to bind to a surface receptor, and they're going to activate another messenger inside the cell. So if we consider our hormone, our signaling molecule from the distant cell, our first messenger, then the enzyme is going to eventually, the receptor is going to eventually activate, often via some sort of enzyme, a second messenger, which will then carry that message down into the cell. So I want to look a little bit more closely at these second messengers and show you an example of how the variety of receptors on the surface of a cell can determine a lot about the physiology of that cell. So let's do a little bit more on second messengers. And for our example, we're going to use adrenergic receptors. So these are receptors that respond to, that are the receptors for the ligands adrenaline and noradrenaline, or epinephrine and norepinephrine. And one of the reasons I like this example is because these molecules can be both synaptic signaling and endocrine signaling, depending on whether they're coming from a neuron and they're signaling within the nervous system, or they're coming from the adrenal gland. And these receptors belong to a class of what are probably the most well-known and well-studied second messenger receptors, what are known as G protein coupled receptors. And you can see that each of these receptors has bound to it a little orange circle indicating the G protein. And the way these work is when the receptor binds its ligand, it releases the G protein, which goes off and usually is going to either activate or inhibit some enzyme which is actually responsible for producing the second messenger. We're going to look at two of these and see how, depending on the type of receptor you have, you will respond differently to the exact same stimulus. As you may or may not know, adrenaline and noradrenaline, or epinephrine and norepinephrine, are responsible for your so-called fight or flight response. They are going to help you respond to a stressor. Imagine, if you will, you're out wandering in the woods, having a good time, and out drops a bear. Roar, scary bear! Ah! You have two options. You can put the Davy Crockett to it and fight the bear, or you can do the sensible thing and run away. But either way, you're going to need your heart rate to speed up, and you're going to need to make sure that your blood flow is prioritized for your lungs and your muscles to help you either fight the bear or run from the bear. So when you see the bear, you're going to dump adrenaline and noradrenaline into your bloodstream from the adrenal glands. And some of those adrenaline and noradrenaline molecules will make their way to the smooth muscle of the arteries going to your digestive and reproductive organs, where you will predominantly see the alpha-1 form of the receptor. So when adrenaline or noradrenaline bind to these alpha-1 receptors, they release a G protein, which stimulates the enzyme phospholipase 3, which converts the precursor molecule, phosphatidylinositol 4,5-bisphosphate, or PIP2, into the second messengers, inositol triphosphate and diacylglycerol, both of which cause that smooth muscle to contract. Adrenaline and adrenaline travel to the smooth muscle of the arteries and arterioles in your abdominal and reproductive organs. There they will find alpha-1 receptors, which will cause that smooth muscle to contract, limiting blood flow to those organs that you don't really need to fight the bear or run from the bear. Those same signaling molecules, when they travel to the smooth muscle of the bronchioles and the lungs, will find primarily beta receptors, not the alpha-1, but beta receptors. Beta receptors use a different G protein, which activates the enzyme adenylyl cyclase, which converts ATP into cyclic AMP, which triggers smooth muscle relaxation. So different receptor, different second messenger, opposite effect. So in the smooth muscle of the bronchioles, we will affect vasodilation from the smooth, not vasodilation, but bronchodilation from the smooth muscle relaxation, allowing you to more efficiently oxygenate your blood. So the same signal at two different cells with two different receptors is going to have the opposite effect. So that's an example not only of endocrine versus paracrine signaling, but also how receptor complement can determine the physiology of the downstream cell. Hope you had fun. Study hard. See you later.